Welcome everyone to the last Discovery to Commercialization workshop. Got a great lineup today to wrap it up for the season. Next slide. So just so everyone knows, I'm David Granville. I'm the host of the Discovery to Commercialization, and thank you all for coming and joining in on us today. Next slide. So the purpose of the Discovery to Commercialization, for those of you who haven't been on before, is to really try and bring the all sectors of life sciences together across BC and get people more familiarized from the academic side of what's going on in industry and vice versa. So at this time, I'd like to acknowledge that this is taking place in the unceded lands of the Musqueam, Squamish, and Tsleil-Waututh nations, and we're thankful that we can be presenting and work, live, and play on these lands. Next slide. So we got a great lineup of speakers. I'm going to allow our chair to introduce the speakers, but first I'll just go over some of the housekeeping things. So this event will be recorded and posted on the website. So if you have any questions, then I encourage everyone to come up with questions so that don't be shy. Please post them on the chat box, and Nancy will ask the questions. And please put your Zoom on mute so that we can't hear you during the talks. Next slide. So at this time, it's my pleasure to introduce Nancy Harrison. So many of you she probably doesn't need introduction. She's been one of the veterans in the BC biotech industry. She was the co-founder and former president of Methylation Sciences. I seem to run into her at a lot of different things, but she's currently a venture partner at Amplitude Ventures. So at this point, Nancy, thank you for agreeing to chair, and I'll pass the virtual microphone over to you. Thank you.
Great, thanks, David. I always think veteran's a kind word for saying old and been around for a long time. So if you're old enough and been around for a long time, you show up in all sorts of different places. But I'm really happy and excited to be here today to talk about precision medicine. I really think it's the wave, and we're going to hear from 4 really interesting speakers today who have come at that topic from a very different perspective, each of them. And we're going to hear all the way from preclinical to clinical development and how to really implement precision medicine. So really excited to do that. As David said, the speakers will each have 10 minutes. Please put your questions in chat. I'll do my best to moderate them. And then at the end, we'll bring all 4 speakers back and we'll have a discussion. So we're going to kick off with Lana Janes, Dr. Lana Janes, who is the CEO of Abdera Therapeutics, which I have the pleasure of working with. So I'm really excited about that. And she's a highly experienced biopharmaceutical executive. For those of you who haven't had a chance to meet her, she used to be the venture partner at Mari Bioinnovations, where she incubated and founded Abdera Therapeutics. I'll let her tell you more about Abdera, but it's a precision oncology company developing novel radiopharmaceuticals. Lana's got extensive experience in negotiating licenses, co-development deals, IP, acquisitions, all sorts of the spectrum, clinical development of companies. And so really a breadth of experience. She holds an AB in chemistry from Harvard and conducted her PhD and postdoc work in chemistry at McGill University. So I'll hand the mic over to you, Lana.
Great, thank you, Nancy. Firstly, I'd like to thank David and the whole team here for inviting me and giving me a chance to explain Abdera Therapeutics to you and the journey that we've taken. So let me just share some slides that I've prepared. So let me see let me see if I can share. Can everyone see those? Yes, okay, great. So I'll tell you a little bit about Abdera. And Abdera Therapeutics, as Nancy said, is a targeted radiotherapeutics company. And we're focused on really building the best-in-class next generation of targeted radiation therapies for patients with the hardest to treat cancers, and those are the relapsed, refractory, and metastatic cancers. and tell you a little bit about Abdera. So just over a year ago, we launched Abdera Therapeutics, and it was, as Nancy indicated, I worked for a few years as a venture partner at Admari Bioinnovations, which is one of the technology venture groups that is pan-Canadian that helps to do exactly this, which is build out companies based upon cutting-edge scientists that have a really competitive advantage in the marketplace going forward. And so that was a lot of fun, and in building that out and finally launching the company. And we had the great fortune to also have as a partner Abdera Biologics as a co-founding partner to help us in our discovery efforts. And I think this brings up our first point, which is in this venue, you're talking about just basically discovery to commercialization. And I think you've probably heard it all year, but it's so critical to have a village of people around you and to bring in the right partners. Even if you have great cutting edge science, it takes more than just a single partner to help develop things. So I would say Abdera is a great example of how partnerships in the industry are so important. And it's not surprising to hear that there's a need for precision therapies, and we're focused on oncology diseases because 1 in 2 Canadians will be diagnosed with cancer in their lifetime, unfortunately. And the conventional therapies to treat those are drug therapies and surgery and radiation therapy. And what Abdera is focused on is bringing something else to the table, which is targeted radiation therapy. So rather than the conventional external beam radiation therapies, which are used in over 50% of treatment of different cancers, we're looking to have really what is injectable targeted radiotherapies that leverage the power of antibody engineering as well. And so what is precision radiotherapy? And all of you are scientists, I'm sure that this will not look unfamiliar to you. And in many different forms of cancer, there are specific tumor antigens that are overexpressed on the cell surface. And so what targeted radiotherapy in a nutshell is designing the correct targeting moieties, whether that's antibodies or small molecules or peptides, that effectively can seek out these unique postal codes on the surface of different cancer types, and then deliver through some really neat chemistry, deliver a radioisotope that can emit either a powerful particle that can help destroy cancer cells, or also can emit particles that allow you to image and therefore show effectively where the cancer has metastasized to. So this is really like a weaponized target therapy. And this is an area that's really been growing quickly as a modality over the past 10 years. And recently there's been 4 targeted radiotherapy drugs approved by the FDA. And to show you what that looks like, this is a great example. Here's the promise of radiotherapy and why everyone should be aware of it as a modality in much the same way that you're thinking about cell therapies and bispecifics and antibody drug conjugates. Don't forget that there is another modality, which is radiotherapy. And sorry, this has moved ahead a bit. Here's a patient who had metastatic castrate-resistant prostate cancer, and you can see it has metastasized grossly throughout their entire body, the abdomen, lymph nodes, everywhere. This patient was treated, the middle picture is after 2 treatments with a targeted radiotherapy called PSMA 617, and 8 months later, you can see that there has been a large retrenchment and killing of most of the tumors that were metastatic. With an additional treatment, even better outcomes. So you can see remarkably in basically a 10-month period, this patient who was at end stage of disease has had a complete remission in their disease. And I think what's important to note is, I've just put a little note down here. This was an academic study out of Germany in 2015. So you can see that this is something that was happening in a professor's lab. And then the next nice part of the story just came out a few weeks ago, which is this drug has now been approved in the United States. And this drug was from that early stage of discovery in a German academic's lab some 8 years ago. The drug was partnered with a US company called Endocyte, and that company developed that drug through clinical trials. And then before even that drug was approved, the company was acquired by Endocyte for over $2 billion. And it was this product that was driving the acquisition. And now you see that the FDA has approved this. And what's really remarkable about this is that this is really bringing solutions to patients and caregivers and physicians who otherwise had no options left for these patients. So the patients right now who would get this kind of drug are patients who have been previously treated, are now no longer responding to hormone therapy, and for which surgery is not an option. So I think this really nicely shows the power of targeted radiotherapy moving forward as a new and emerging modality. Now, you all probably know that radiation therapy is not new. And in fact, there's been targeted radiotherapies. I think the earliest one was probably 50 years ago that was targeting thyroid cancer to deliver radio emitting iodine tracers. But the important thing is right now, with the confluence really of the power of molecular biology, of being able to really design highly selective, highly specific, high internalizing antibodies and cyclic peptides and the like that can really seek out that unique postal code on these unique antigens that are being expressed, and then combining together the power of advances in linker chemistry, conjugation chemistry, and advances in nuclear chemistry, and the ability to produce some of these highly effective medical isotopes at a scale that means it can be commercialized. I think this is why the time is now for radiotherapy. So I know a lot of you think about when do you translate from discovery to commercialization? And what I wanted to discuss with you today, and just give you this snippet of a bit of a history of why we think Abdera is uniquely positioned with our unique approach to be able to develop new therapies for other hard to treat cancers out there. The unmet need is so high, and unfortunately there are so many cancers that remain untreatable, that the timing is really now because of a confluence of all of these developments. And not surprisingly, this science is translating extremely well and is highly desired right now by investors and companies who are looking to say the time is now to translate this. So I guess that I wanted that to be my take-home message, that a lot of you in the academic side are doing cutting-edge science, and it's really finding a community that can help you develop and translate your technology through to find the right partners, the right investors, and to really ultimately get that into patients. So I think what I wanted to really stress upon you is you really need a continuum of experience around you to help build out your technology. So there's lots of folks in our community who are helpful. There's lots of investors like Nancy and Amplitude Ventures and other groups, Admare Bio Innovations and the like, to really help you translate your really cutting edge academic science. And they've all played a big part in the role in founding Abdera Therapeutics, which I have the real honor and pleasure to be helping to build out right now.
Great, thanks, Lana. I know we have a couple of minutes just because we were a little ahead of schedule. So, I thought I might ask you a couple of questions before we move to Handel. I think what hopefully isn't lost on this group is it's quite fascinating to see the engineered approach. Those of you who've seen me speak before, one of the things, I'm an engineer by training, and what I love about a lot of these sciences is this is an engineered molecular approach. Here we're putting a biologic with a small molecule with a radioisotope. And as you said, we're really developing sort of a killer, almost like a killer app for cancer with this one. Maybe you can talk to a little bit of the competitive advantage in Canada. that we have just
Sure.
because of our history, just as an interesting thing that people might not have thought about around radiopharm.
Yeah, no, that's a great question. You know, when we started looking at this at Admare Bioinnovations, when I was a venture partner there, the reason why we were attracted to this area was because of the really the strong excellence in Canada, and in particular in Vancouver. When you think about, there's many different disciplines that are in these constructs, as Nancy said. You know, you have chemistry, you have biologics, and you have radioisotopes. And when you look around Canada and Vancouver in particular, those are all of unique and great strengths of ours. So Canada has had a long history of being at the forefront of nuclear medicine and creating nuclear medical isotopes, certainly through the days at Chalk River, even 40, 50 years ago. We have Triumph here in Vancouver. We have McMaster and Center for Probe Development and Commercialization. So globally, Canada is recognized as being really a leader in the production of medical isotopes. And because of that, we also have a lot of clinicians who are really key opinion leaders and are used to treating their patients, who understand how nuclear medicine and nuclear imaging fits in the paradigm of treatment, particularly in the oncology spectrum. And so you have clinicians who understand and can bring that technology to their patients as well. And then, you know, you have in Vancouver, for example, Abdera is one of our co-founders, and you look at their world-class antibody discovery company. There are several world-class antibody companies that have been hand-built in Vancouver and are recognized worldwide as really being able to work with partners to, you know, build out these bespoke, highly engineered biologic molecules that need to have specific properties that you're looking for therapeutics. So all of these pieces really came together, and it's that continuum of knowledge that was naturally here that made it really a no-brainer for us to say, you know what, let's start building something out. And then we spent quietly and methodically, we spent a few years building all of the relationships around us. And that's relationships with the doctors, with the nuclear medicine experts, with the radioisotope manufacturing experts, with you know, investors in the space, and having them really understand, here's our story and here's what we want to do. And we found there was a whole lot of support when we actually wanted to launch the company and do that. So the relationships, including like the ones you're making today, are really important. Getting your message out and saying, we're playing to our strengths here and we know this space well.
Yeah, that's great. Great feedback and a great talk, and some really great points in terms of thinking about this. The people who are on the call, you know, I think we would all say it really does take a village, and there's a lot of us that are around here to help support the ecosystem. So don't be shy and ask for help. We're all of us who are doing this oftentimes get that. Well, thanks very much, Lana. That was a really informed talk. Really appreciate it. So now I have the pleasure to introduce Handel Kim, who is back on video. So that's good. I'm glad to see him. Handel is the co-founder and CEO of Variational AI, an AI discovery company based here in Vancouver. Variational is applying their state-of-the-art generative AI to discover novel drugs for a range of different diseases. And I'll let him tell you about that. He's started 3 companies and brings over 20 years of executive management experience and leadership from venture-backed startups and publicly listed companies in Silicon Valley, Canada, and the Asia Pacific. Handel also does a lot of community work and is on the boards of AI Network of BC, the Digital Supercluster, the United Way of British Columbia, Lower Mainland. So I'll turn the mic over to you, Handel, and you can tell us a little bit about variational and precision medicine in AI.
Thank you very much. Let me share a screen. And assuming everyone can see it.
Yep.
Thank you very much, Nancy, and it's a great honor to be invited to speak and join such an esteemed panel of speakers. As Nancy said, I'm co-founder and CEO of Variational AI. We're a Vancouver-based AI for drug discovery companies. And I thought I'd take a little bit of a whimsical sort of view and talk about what we've been doing. And specifically, the ask was around what we did around COVID-19. And so A bit about the company. We were founded in September 2019. We're based here. We've always been based here. We're a small team, but the leadership team has a proven track record in both machine learning and drug discovery. We are machine learning scientists from some of the top academic and corporate organizations engaged in AI machine learning research and development, as well as an extensive track record of drug discovery success. We are venture backed, and we also have non-dilutive funding from the Canadian Digital Technology Supercluster in partnership with Admare and the Vancouver Prostate Center and UBC, which I'll be talking about a bit more later in the presentation. What we do is, as Nancy mentioned in the intro, is we're building a drug discovery engine, that will rapidly generate novel and optimized molecules for unmet medical needs across disease areas to hopefully redefine the economics of drug development. And that's our big, hairy, audacious goal. But before I kind of dive into what we do and what we did on this project, I wanted to give a very high-level primer on machine learning and AI. And I know everyone here is extremely accomplished in the sciences, but sometimes, it's good to take a 30,000-foot view about what AI is and what it isn't. So in this case, what we're talking about is the traditional types of machine learning that you're probably familiar with. These are known as discriminative models. And in this case, the example given is that, let's say you train a discriminative model on a bunch of paintings by Van Gogh and a whole bunch by Picasso. And you train it with thousands or hundreds of thousands of samples of these paintings. And then what happens is the AI, or the machine learning, is then able to differentiate and classify such that you train it on this model, on this data, and then you give it a new sample, a new painting, and it's able to classify with very high accuracy that this is in fact a Van Gogh painting, and so it is. And you're probably familiar with these canonical use cases in things like medical imaging and in radiology, et cetera, where you train a machine learning model, a discriminative model, on a whole bunch of scans that are negative and hopefully a whole bunch that are positive. And then you give it a new image and it's going to be able to tell you with very high accuracy whether it's negative or positive. And so luckily for this person, it's negative. So already these models are being used. They have good accuracy. They're being adopted at scale across multiple industries, such as in automotive autonomous driving, in medical imaging, obviously, in digital media, also in e-commerce, etc. And this is in full flight. And that is the machine learning most people talk about when they talk about machine learning. There is, however, another type of machine learning known as a generative model, where the task is not to classify an input, but rather is to learn what makes that input, and then is able then to generate novel samples that it wasn't explicitly trained on. So in this case, for example, here's another Van Gogh painting, Starry Night. Assume that we're able to train on a whole bunch of Van Gogh paintings. And this model effectively learns what makes a Van Gogh painting a Van Gogh painting, and then you give it another image, and it's able to then turn it into a Van Gogh painting. It is able to understand the elements of a Van Gogh painting and then generate novel instances that it wasn't trained on. Now, this is known as a generative model and is less commonly used because it's far more difficult and far more advanced. What we effectively do at a very high level is we use it for molecules. We train this on molecules. The generative model understands what these molecules are and then generates novel molecules. And please, these structures are obviously not correlated, but we haven't had time to go to the graphic designer. But what we do is we use this generative model to generate novel and optimized small molecule therapeutics against a given target. And so we are not a discriminative machine learning, which is about 95% of all the AI for drug discovery out there. In fact, we are generative. So what are the value propositions of generative machine learning? Well, number one, they deliver novel chemistry. Using a generative model, you're able to efficiently explore unexplored areas of chemical space, and it's not through brute force screening. The second value proposition is that we're able to do multi-parameter optimization. We're able to optimize multiple properties, drug-like properties that you want against a given target, all at the same time, and therefore speed up the entire process. Very quickly, the status quo versus the generative machine learning is that if you look at this, this is a representation of the grid on the right. On the right, you'll see a bunch of molecules, and they look fairly structurally similar. The values underneath are true docking scores. And you'll notice that there's quite a bit of variance in terms of the scores. And this underlines the fact that molecular similarity in terms of structure is not necessarily uniform. So you're going to have a structure that's very similar, but will have a very different property, in this case, a predicted binding affinity, than another. So these colors correspond to the predicted docking scores. And you'll see here a jagged landscape. And this, what happens is, in traditional drug discovery, you look at potentially a starting point, and then you look in areas close to it. And then what you do is generally you look around and see if you can find novel structures and scaffolds, and you do a search in that local area. But that local area most likely is not able to deliver the true properties that you want that would make a very, very effective drug, and also across multiple properties. What we do essentially is we search not by structure, because chemical space is massive. It's on some estimates 10 to the 60 molecules. It's chaotic. You know, the structures that we use to interrogate chemical space are not necessarily the best way to do it. Small structural changes can induce very large property changes. And so it's very difficult to brute force search chemical space. What we do is we take this, we transform it using our generative model, and we train the model then to generate novel molecules that it wasn't trained on. What we do is we project into a structured search space where the empirical properties of interest that we're looking for, whether they be selectivity against a target, no activity against a non-target, pharmacokinetic properties, potency, etc. These change smoothly. This is state-of-the-art machine learning, which takes a discrete representation in molecule space and turns into a continuous representation, where then optimization can be done very smoothly and efficiently. So essentially what we do is a very different approach to drug discovery, and a very fundamentally different approach to what chemical space is. So stepping back, wow, okay, we had this great technology, and we were in a situation in 2020 where we would like to validate. And we had a proof of concept thanks to the fine folks at Edmari, and especially Lana James, thank you very much. But when coronavirus hit, it was an opportunity where we wanted to contribute. If you've got a machine learning platform that is supposedly able to rapidly discover novel drugs, well, you want to have the most impact as possible. And so when we looked at COVID-19, we thought, what a great opportunity. There's a lot of data, or there's a lot of activity, there's funding behind it, and obviously there's urgency. This was and still technically is a very large unmet medical need. The problem is that there was no data, because it was a novel coronavirus back in 2020. And the thing about machine learning is we train on data, and specifically we train on activity data. If there's no activity data and we use machine learning, how the heck are we going to build a model to generate new molecules? Well, we looked at the actual target, and we realized that there was a tremendously large overlap between the main protease of SARS classic, SARS-CoV, as well as SARS-CoV-2 and COVID. And so what we realized is that the active site was also conserved. So we hit upon the bright idea, and of course we weren't the only ones, to use SARS classic data, of which there was a lot, because we stopped developing therapeutics back in 2005 when SARS went away. There's a lot of it publicly available. And so what we did was we applied a method known as transfer learning, where essentially we used data about SARS-CoV, SARS classic, and the main protease, and we were able to train on that and then apply it to SARS-CoV-2. And as we were going forward, there'd be additional data that would be published on SARS-CoV-2, because this was a massive international effort, and we were able to avail ourselves of it. The fascinating thing was that we found actually tremendous improvement in the use of transfer learning and training on the SARS-CoV data versus just training on SARS-CoV-2 data. And so this was a great revelation on us. So we thought, well, where do we get this data? Well, we had to go all the way back to 2003, 2004. And if you'll recall, this is Hong Kong, this was SARS classic, and these are just some touchstones from back in the day. I just thought it would be interesting. If you don't know, young people here, this is Paul Martin. I think he was Prime Minister of Canada for about 3 years. In any case, going all the way back and using this data, we were able to build and construct a very powerful model that is able to generate results now. And where are we today? Well, we generate hundreds of novel and optimized molecules and compound structures with very high predicted binding affinity, high selectivity, good synthesizability, and other properties of interest. The transfer learning is working, or at least it's working so far as we've told. as we can tell. We're waiting for our molecules so we can test them in assay, and obviously we've been impacted by the Russian invasion of the Ukraine. In terms of our partners were there. I mean, everyone seems to be working with the same partners like Immune, et cetera. And so that is materially impacted. However, we're happy to announce that work has continued, and we are going to be publishing results as soon as we're able to get them. The great thing, though, is that we're now taking this approach and we're going to apply it in oncology. So stay tuned, because this is just the first chapter. And that's my presentation. Thank you very much.
Great. Thanks, Handel. I think one of the things that you kind of glossed over, but given that we have a couple of minutes, you talk about your tool. I think, when we first met, I said, well, that's all a great tool, but you basically told me I've been doing my job, or, the medicinal chemists have been doing their job incorrectly the whole time. So maybe you could talk a little bit about that learning and the leaps of faith, and how one needs to sort of think about that in the continuum of a village.
Well, so you may have confused me with someone else, but I hold medicinal chemists in the highest regard. Absolutely, except maybe Jason Crawford, but that's only because he's here and I know him, but I'm joking, of course. Look, the fact of the matter is AI is not going to replace medicinal chemists for decades. So everyone who's already got their PhD are probably okay. I'm being a little flippant here, but the fact of the matter is machine learning is based on the induction and the absorption of human knowledge. The concept of human in the loop to help train a machine learning model or an AI is absolutely integral. And so we ourselves have internal medicinal chemistry capacity. We use that in order to inform the model, because machine learning at the end of the day is extremely literal. It doesn't care. So it needs that human intuition to guide. And we've had tremendous benefit and improvement from the practical knowledge of medicinal chemists who've informed our discovery, because we'll come back and go, look at all this. Aren't these great? And the med chem will go like, these are just pains, oh, we didn't know that. So you go back and you learn and you get smarter and smarter. And the better the interaction, the more frequent the interaction with a medicinal chemist, the stronger the model is, because at the end of the day, what we want to do is be able to incorporate the intuition that a medicinal chemist has through their extensive years of training, through the scholarship and research and scars, and then integrate that into the model and build a better model. So really what we're doing is, imagine if you had this super awesome medicinal chemist who could be trained, for millions of years and have that combined knowledge. That is one way of thinking about what we're trying to build.
Yeah. Jason, we're going to hold questions to the end, and then I'm for sure going to come to you first. So because I'd love to hear, but I'm gonna switch from Handel to Robin just to sort of keep us on track, and then we're going to have a full discussion. So I won't forget. So I now have the pleasure. Thanks very much, Handel. I appreciate that conversation, and I'm sure we'll have more of it. We're now going to switch a little gears from talking to companies that are preclinical to speaking to Dr. Robin Sharington. And Dr. Sherrington is the Executive VP Strategy and Innovation since March 2019 at Xenon Pharmaceuticals. He's previously held many other positions there: Executive VP, Senior VP, and Vice President of Business Development and Corporate Development, and many other roles. And he's been with Xenon since 2001. Prior to being at Xenon, he worked at Pfizer as a neuroscientist, and also he was director of neuroscience for a few different companies, Axis Pharmaceuticals and Sequana Therapeutics. And Dr. Sherrington has a postdoc fellow from U of T and received a PhD from the University College London and his BSc with honors from the University of Reading. So really welcome, Dr. Sherrington, and I'll turn the screen over to you to tell a lot about a clinical precision medicine play that Xenon has. And you're on mute, Robin.
Thank you. That's a classic mistake for you.
Yeah, somebody had to do it.
Okay, can everyone see the slides okay?
Yes.
Great. Let's see if that's coming up. That should be better. Well, first of all, thank you so much for the opportunity to speak to you today. What I thought I would share, given the title is around precision drugs, is probably some of the work Xenon is undertaking, developing Zen496 as a phase 3 precision medicine for a rare genetic disease called KCNQ2, developmental epileptic encephalopathy. And sometimes you hear people talk about precision medicines, perhaps from a population basis, where there's certain variants in the human genome which might predict adverse drug reactions or might predict someone who's more likely to respond to a treatment from that population perspective. But this is really a discussion around targeting the root cause of the mechanism of the disease through a drug. So it's a true precision medicine strategy targeting this root cause. One of the things you have to do, and I want to use this example just as really some of the steps you need to take while you evaluate and think about such an opportunity. So one of the things you need to do for early is to really understand your genetic disease and how it works. And one of the things which is important about KCNQ2 DE, which brought us to this indication, was there was a degree of severity in the disease. And that degree of severity in the disease was highly correlated with the impact that the genetic mutation had on the function of the KCNQ2 gene, which is translated into the potassium ion channel Kv7.2. The greater the loss of that ion channel activity, or the KCNQ2 current, the greater the severity of the disease, whereby relatively low loss of activity, the children would actually have a fairly favorable outcome with some early seizures in life, but they wouldn't suffer from any developmental consequences, whereas the greater loss they would. So theoretically, if you had a drug that targeted this gene and you could actually replenish some of the missing current, you could imagine that the child might have a better outcome longer term. And this really is the hallmark of a precision medicine strategy for this disease. So in the context of the DE form, the developmental epileptic encephalopathy form, universally poor prognosis, seizures are very difficult to get under control, developmental delay or arrest, moderate to severe intellectual disability, they don't form language. So you can see there's tremendous high unmet medical need in this population. Even though it's quite small, the medical need justifies the development of a treatment that's precisely looking to manage these manifestations of the disease. It's very important that you learn as much as you can about the disease, either through academic research that may be underway, or you sponsor such academic research. Because the more you know about the natural history, the better you are at being able to design the appropriate clinical trials, the clinical endpoints, and understand how your therapy might provide benefit against the burden of disease. And so we're fortunate that in this, for this particular disorder, natural history work has been done, and they've looked at the performance of these children outside of their seizures, which need control, but they've also looked at the other factors of the disease burden, and they've looked at the performance in functional domains such as the ability to walk unaided or walk at all, fine motor skills, pick up toys, play with the toys, feed themselves, being able to speak and communicate. And you can see from the numbers here that these children are severely disabled. So it really talks to the high medical need, which provides the commercial opportunity to develop a drug that targets the root cause of the disease. The other thing you need to understand is actually what the gene does. I think this is very important, particularly when you're thinking about developing a precision medicine. Unfortunately, in this case, because it's a potassium channel, the KCNQ2 potassium channel function was known. It's one member of a gene family. It controls the excitability of a neuron. So you can already imagine if that excitability is missing, if that gene is missing, you're going to get an increase in the excitability of neurons in the brain, and that can lead to seizures, but it also leads to the other consequences of the disorder we just went through. And understanding the fact it's loss of function, not gain of function, is very important, because it teaches you what therapeutic strategy you want to take to develop a precision medicine such as this for these children. And so 496 uses an active ingredient. This is actually an active ingredient that was previously developed and commercialized by GSK as an anti-seizure medicine. It was removed from the market for commercial reasons, but it is uniquely positioned as a drug because it actually targets the loss of current that occurs in the KCNQ2 children. And it targets the KCNQ2 3 heterotetramers, which form the major potassium channel that's disabled through the genetic mutations in KCNQ2 for this disorder. And what you can look at, and this data was published, is you can look at the ability of such a drug as a precision medicine in the context of the mutations that cause the disease. If you can measure the potassium current, as can be done, you can see that the drug is able to replenish the missing current in these mutated channels. And this really underpins the precision medicine strategy for this orphan genetic disorder. And the other thing you can look at is we were fortunate in this case that a number of physicians had been using the drug before it was pulled from the market to see if it would work in these children. This was off-label use, but they actually demonstrated in these children which had failed to respond to multiple anti-seizure medicines that had been previously tried, had a very good response when they put the precision medicine on board on top of these anti-seizure medicines in these children. And you can see 11 of 11 of the patients who were seizing had a very decent and very good response to their seizure control, from seizure freedom to greater than 90% reduction. So again, this is addressing the seizure unmet medical need in these difficult to treat children. And obviously, if you leave seizures untreated, the children have the risk of unexpected death as a consequence. This is a very important part of their medical care, is to get the seizures under control. And the other thing they did, because this was a precision medicine, was to look beyond just the seizure control. And was there any evidence that this precision medicine was also helping out perhaps with some of the developmental aspects I spoke to earlier in these children? And they actually reported in 12 of 17 patients that certain psychomotor development was improved. So again, this talks to the advantage of a precision medicine that's targeting the root cause of the disease. And finally, one way you can look at this in the context of epilepsy is for the epileptic encephalopathies, there's interictal disorganization in the EEG. So you can see on the left-hand side in a patient, this was a highly refractory patient, and you can see the EEGs become much more organized in the presence of zogabine. So this is talking to the non-seizure effects of replenishing the potassium current. And they reduced the dose in this child, and you can see there was an incremental increase in the disorganization. So again, it is supporting the utility of this drug in these children as targeting the root cause of the disease. And before you take on the endeavor, you really need to understand the size of the commercial opportunity to warrant the investment you're going to make in the development. And really, this starts with understanding the prevalence of the indication. And again, we were fortunate in this context. You would either have to sponsor this work, but in this context, there was a large study done in Scotland where they genetically typed all children with seizures between up to the age of 3, and they're able to give us a birth rate of 1 in 17,000, of which about half of these subjects have the developmental epileptic encephalopathy form where the high unmet medical need is. And so you can see this is actually a reasonable birth rate and a reasonable number of patients to warrant the investment you're going to make in development. And furthermore, further genetic work, which we've been sponsoring with some of the companies that do the genetic testing, One of the issues you have with these genes, which are recently cloned, is you get a large number of variants of unknown significance, where there's a rare variant, but they can't say it's causal. You can sponsor work such that you can actually address some of these variants and recall them as causal and in the sense pathogenic and causing the disease. And we've done some of that work, so these numbers will grow by 20%. In the context of an ultra-orphan disease and ultra-orphan disease pricing, this is a significant increase in the opportunity, the commercial opportunity. One of the challenges, though, is you've got the prevalent number. That does not mean everyone's been diagnosed, because there is restrictions in how broad genetic testing for genetic disorders is available. And this is some work which was done in the US. And so we got together with one of the genetic providers and 3 other companies and sponsored genetic testing with the sole purpose to actually diagnose and increase the diagnostic rate in these children to help support our eventual commercial opportunity for the product after it's developed through its phase 3 program. So you have to think about these ways of getting the diagnostic rate up higher so you can capture more of the patients with your precision medicine. And sponsoring genetic testing is just one of the ways we approach this. And it's been a very, very successful and well-received program, particularly in the US, where genetic testing isn't always available through their medical insurance. You also have to understand how the patients are currently treated today and what is their journey and what's the physician paradigm for their treatment. So you can begin to imagine where your precision medicine can have an effect. And the current treatment for these patients is to use standard neonatal phenobarbital levetiracetam, standard neonatal anti-seizure drugs. These 2 drugs actually do not work well for the babies at that time. And then it becomes a bit of a hit and miss where you try one of several different ASMs to see which works best. But through conversations, through primary market research, you can then introduce to physicians your target product profile for your precision medicine and see how they would how well it would be received and how they would use the product. And that gives you an opportunity then to model the commercial opportunity. And obviously for this particular medicine, given the high medical need, it was well received from the pediatric epileptologists who are the primary care for these children, that they would really want to use this as early as possible after the genetic diagnosis, which was why it was important to sponsor programs to get that genetic testing done earlier and earlier to help support not only identifying the children earlier, but they can get access to your precision medicine earlier so you can have a hopefully greater impact. The other perspective is to take advantage in the precision medicine orphan rare disease space. A number of countries have orphan drug acts, and that enables you to take advantage of more regular interactions with the regulators, the FDA or the EMA in particular. And it also gives you more faster regulatory interactions, so they respond to your questions or queries or give you information more quickly, as well as they give you some market exclusivity provisions as well before a generic could come into the market. All of these are extraordinarily helpful, and one of the things you need to do early on is get into discussions around what your development plan should look like, to get their buy-in to that development plan. And often with these orphan diseases, the development plans in terms of scale are very, very different. And for this product in particular, both the EMA and the FDA agreed to a single phase 3 study of 40 subjects, which would be sufficient for filing an NDA or an MAA for marketing approval of the drug in those 2 jurisdictions. So it was very good to have that information early so you could look at the scale and the cost of development, again, to basically justify the investment you want to make to realize the commercial potential.
Robin,
so-
sorry.
yeah.
Oh, you may be at the end. We're just
I'm
running a little tight on time.
I'm right at the end. So in summary, we're developing a precision medicine that's targeting the root cause of this of the disease. We're able to get into advanced development quite quickly, and we believe because this will be a precision medicine in these children. There isn't one available today. This will be the mainstay of treatment for these children once it's available. Okay, thank you.
Great, thanks, Robin. Just to keep us on track, we'll stave off questions until the 4 of us or 5 of us are all together. But thank you very much. Really interesting, very different stage program, so it'll be interesting to see the questions. Now I have the pleasure to introduce Dr. Cherkasov. And Dr. Cherkasov is a Tier 1 Canada Research Chair in Precision Cancer Drug Design and has been a really meaningful drug developer in our community in Vancouver for quite a long period of time. He's a professor in the Department of Urological Sciences at UBC Faculty of Medicine and is a senior research scientist at the Vancouver Prostate Centre. His research interests include computer-aided drug design, so we'll talk a little bit about that and the crossover, and then drug profiling, new cancer therapeutics, and development of CAD tools and applications. And before I turn it over to Art, I'm just going to remind everybody to please feel free to start posting questions in the chat, and we'll gather them up for after Art's finished his presentation. So over to you for about 10 minutes, Art.
Okay, thanks very much, Nancy. So before I begin, I'll just probably will make a proper remark that building on what Handel was saying, that computers will not likely replace chemists. But as Jerry Klaus, one of the visionary medicinal chemistry, said on his blog, machines will not replace chemists, but chemists who use machines will replace chemists, those which don't. So, and that's more or less what we're trying to do here, to bring AI, bring computation into life science. And I'll give you just very recent updates on what's been going on in the lab, mostly in regard to also COVID research, because one thing I should mention, that COVID really brought up a lot of science to the frontier and really accelerated the use of AI because of the time constraints and urgency and need. So that's where AI really started to shine. So my lab works in the area of computer-aided drug discovery, and we've been doing it for probably close to 20 years. And the standard workflow with computational drug discovery is the following: that we look at the protein typically, which is a drug target, and we examine its surfaces, its constituents in very, very fine details, looking for potentially binding sites for small molecules. And those small molecules in principle become future drugs once they develop. And so that's the workflow we've been using for decades. Nothing's been changing for years and years, and that was working just fine. So the average success rate of drug discovery campaigns or hit discovery campaigns backed with computers is anywhere from 5 to 20%. And that's compared to 0.03% of a sort of a random wet lab search, which was more conventional before computers became a norm. So that's how it was going on for decades. That's how we've been developing drug after drug. And the workflow was typically with the docking, that's the process I just demonstrated, would involve about 1 to 10 million molecules. So we would take a database of compounds, 1 to 10 million, those which can be made, can be synthesized readily and don't cost much money. And then we would run this docking and we would identify anywhere from 100 to a few hundred compounds, which then get passed to the wet lab, and then wet lab screens them and finds the actives. Again, that's been going on. It was working fine. And that's the approach we've been using back in the days, back in the years, including the now famous story when we just used 5 million molecules and we went through docking and other in silico tools, and we identified a drug which was then licensed to Roche, which was and still is the largest academia licensing deal in Canada. So we licensed it for 142 million US. And that was, again, just 5 million molecules. So it worked. And things really changed about 2-3 years ago, because what happened in about 2018, 2017, the number of compounds just skyrocketed, compounds which are available and synthetically tractable. So all of a sudden, we're facing not 5 million molecules, but we're facing close to 40 billion molecules. That's what's available from vendors these days. And just like Handel also mentioned, but the real number is 10 to the power of 60, or anywhere 10 to the power of 60 to the power of 100. That's what's possible. And that's more than atoms in the universe. So combinatorial problem with chemistry is such tremendous. But even these days, we do not have means to go for 40 billion molecules. Even though we have supercomputers, we have pretty decent computational clusters at UBC. I have my own clusters built over the years with different sorts of funding. Again, that would take us really a couple years to go through 40 billion compounds using pretty sophisticated, powerful machines. Previously, 10 million, that would be typically a couple months. That would be our ballpark time frame. So it wasn't undoable anymore. So that's when we decided to change the practice. We decided to change the way things are done. So instead of working with three-dimensional structure of a molecule, or here it's simplified in 2D, but docking is really a three-dimensional phenomena. It's a kind of Kino law principle. So it's the docking programs run things in 3D, in virtual reality. So instead we said, what if we go from three-dimensional object manipulation to what's called fingerprint? It's basically digitalized zeros and ones string of zeros and ones, pretty standard for AI, and we would feed it into AI. So rather than doing physical modeling of how things fit, we would train a neural network, AI algorithm, to understand how it works in digital zeros and ones world. And so basically we put a little module on top of a regular docking pipeline, which starts learning how things work and how they don't. So as the docking goes in 3D, but it's a very small fraction of a database we need to really dock, this module stays on top of it and learns how things go into yes or no. And more it goes, more fast it learns and starts accelerating the process, or augmenting the data flow. And so as a result of it, we were able to make it 100 times faster. So all of a sudden, we can process not just 10 million molecules, but we processed 1.4 billion molecules. That was the first COVID research. And so when the outbreak, when the pandemics happened, we looked at very quickly, structural biologists resolved all critical proteins in the virus. And so just like many others, we look at the 3CL protease or main protease as the target there. And we passed 1.4 billion molecules through this, what now it was called deep docking algorithm. So docking accelerated with deep models. And that was a game changer, really, because the previous biggest in silico campaign was 130 million molecules from UCSF and was published in Nature. And it took them a while. For us, we went through 1.4 billion in a week, and we published as early as in February of 2020. So this campaign resulted in a paper. So the preprint was in February. The paper went out in March 11. It's probably, these days, probably 20,000 papers on COVID. Our was roughly number 2 and number 3 of all research papers. It's just because this use of deep docking, again, speeded things up so tremendously that after just a month of work, somewhere in February, we published 1,000 molecules, which we thought would be useful for COVID. And so just to verify that, at least several research groups went into that 1,000, we made publicly available, and they have been validated. So they see hits there. And that paper was and still is a blockbuster, has been cited close to 350 times in 2 years. So very highly cited paper. And ourselves, we also went through close to 600 compounds. So we actually purchased them and tested them and found about 30 active. And not to mention, really changed the way actual screening or docking is changed. So ever since, the entire world is trying to do the same. Few papers followed. So the OpenEye campaign, this is the biggest computer in the world. They used at the Oak Ridge Computational Center. They threw in 45,000 computers, GPU cores, to run the docking of the same 1.4 billion. Of course, it took them 1 day with 45,000. So that was the European consortium. They threw in close to 28,000 computers, also to do the same in 1 day, that 1.4 billion. Because we showed them it's possible, so now they just brutally forced that. The closest academia kind of research, they also used, I think it was UCSF as well, they used 8,000 computers to do it in 4 weeks. But we've done it in 5 weeks with just 4 computers, pretty much, and just pretty one person sized cluster. It was a pilot study. So you see the difference, because those guys didn't use AI acceleration, we did. So we figured, okay, fine. So now 1 billion is doable, but we can do better than that. So we went through 40 billion. So we went to vendors and they asked if they can send us larger databases, and they did. So Inamine sent us 40 billion molecules, and we docked them, and we docked them in 5 different ways. So one thing AI really enabled us is to go into these ultra large chemical libraries, but also AI enabled us to go in multiple different ways. So I'm not going to go into many specifics on this matrices. Basically, we started developing voting protocols when one program agrees to another with another with another. So we started developing up to 28 different strategies based on different types of consensuses, because we processed these 40 billion molecules in 28 different ways. And so these were 26 hit lists. Also, we used 2 expert hit lists when just a person was picking up, compare how that works compared to computers. And we ordered close to, I don't recall it exactly, probably 1,400 compounds. So we ordered close to 1,400 compounds, emerged from 28 different strategies. And we started looking which consensus protocols delivered what. Because our also goal was not just to brutally force with AI this enormous database. Also, we wanted to do is elaborate different consensus protocols to automate the process as much as possible. Because these days, drug discovery in silico stage is really expert-driven routine. At the end of the day, there is a person going through whatever computer predicted and picking up what looks good, what not. Medicinal chemists really use their insight. And this is subjective. And this cannot be really generic. So we decided to evaluate all kinds of different consensus protocols to see if we can really fully automate it. Because when 5 programs agree, most likely we don't need expert insight. And so we evaluated these 28 protocols. We ordered 1,400 compounds, and we put them through the standard these days 3CL kit assay. So we used the control, we used all kinds of kinetic measurements. And so we identified 162 active compounds. So this is, again, I was saying, computers give you anywhere from 5 to 20%, what's called hit rate, success rate. So our number was right in the middle. So about 10% of all compounds which were screened were in fact active. And again, more importantly, some of these protocols are fully automated. So that's how we really changed the game. So again, these days, everyone is doing it in the very same fashion. So deep docking is now the way to do it. It's also what I call democratized drug discovery, because now research groups all around the world don't need supercomputers. They can just do it as we show on 6 GPUs, 40 cores. It's basically, $10,000 worth of equipment. And now with that, they can do it also in a very automated way. So you don't need a special human expert to do the hit calling. Again, you can use these consensuses. Again, this type of a workflow when you go and filter and filter and filter. Basically, I have a couple minutes just to give you insight. The blue color means actives, right? So you can see we start with just rough docking, which is roughly contains half of good ones and bad ones. And we keep filtering with different more routines. You see things get blue and blue and bluer. So basically, these are very digested, very processed hits of compounds. So we ended up with 40 molecules picked up out of 40 billions, but you see they're all very blue, very active. So this basically the workflow AI enabled us to do. And so we identified a whole bunch of experimentally confirmed COVID inhibitors and 3CL protease inhibitors. Some of them are pretty far now in preclinical development, so hope is we're going to finish it, and sometimes later this year or the next year, we can even probably get them into some late stage trials or something like that, depending how things unfold. But again, the workflow really worked. We built together a very good group, and with crystallographers, medicinal chemists, virologists, all kinds of talented people. Again, we had tremendous support from all kinds of sources for what we've done. And now, we're going to try to use the same pipeline, same AI-enabled workflow for all other kinds of targets, not just COVID. Hopefully, we'll go back to cancer, because that's where I used to work for before COVID came to life. And so we hope that we'll keep enjoying the same success in all other fields. So we'll be happy to answer your questions later. Thank you.
Great. Thanks, Art, for that overview. I mean, I think I'll ask, well, I'm asking Robin and Handel and Lana to come back on the screen so we can have a Q&A session. I think one of the things that I'd kind of start with of all of you is, 20 years ago, well, first of all, one of the great expertise that I think we underestimate here in Vancouver is compute, and how long people like you, Art, have been doing compute, or Zymeworks is based on that premise as well. Really, what we're all trying to get to is a better hit rate, better drugs, right? So we're holding out the promise of precision medicine. We've been talking about it for a long time. I guess I'd ask each of you, and maybe starting with Lana, a little bit of a question that David posed, but tilted more towards precision medicine, which is, are there any areas for improvement in the ecosystem? Maybe talk about precision medicine in our ecosystem and how we can all help each other. Each of you have talked about how that works. Robin, from a deep expertise of understanding the disease, Lana from engineered molecules, AI, and CAD design. So maybe each of you can touch in your own areas. How do you think we can help each other a little bit better? And what else do we need in our ecosystem? Lana, maybe start with you.
Yeah, I think this, it's a great question. I think it strikes back to what I think my closing comment was, that, drug development now needs a continuum of expertise. And if you're not using that continuum of expertise and looking at, machine learning and looking at very sophisticated bioinformatics, and, you're probably not developing a 21st century drug, and you're going to be, it's not at the cutting edge of science. So when I think about, we were lucky when we were starting Abdera Therapeutics that this partnership where we leveraged Abdera and we leveraged Advare, and but I think that's absolutely right in terms of how we think about, a lot of times we think about our partnerships in another country, or we think about elsewhere that we can go. I think that within Vancouver, there's so much in terms of AI, machine learning, antibodies, and I think the power of what really is exciting to me is the power of AI to be able to design bespoke antibodies in silico and CDRs in silico, perhaps, as opposed to some of the actual experimental work that is done through, immunization campaigns, phage display, and the like. I think that it really will be important for us to work close together. I mean, Handel and I worked together when I was a venture partner, and Art and Handel and I worked together, and we are a very small community, but a really sophisticated one. And a lot of times we look beyond our borders or beyond our own community to be able to work together and actually start thinking about how we can work more closely together with the people that we know, because when people, when you're working with somebody, if they have the funding, if they have the relationships, good things will come from it. And so I guess that's what I would say. But what I'm really excited about in the future, specifically for Abdera, since we're always looking for unique biomarkers and unique tumor antigens that we can leverage our platform around, I really think there's an opportunity for us to partner with like the very best in bioinformatics and artificial intelligence, and some of the chemists in our community as well, and protein engineers to kind of help us build the next generation of what's going to be in our pipeline right now. Yeah. So Handel, we need you to focus a lot on, you just need to solve that problem of applying AI to CDR sequences. And if you could do that next month, I'd be really happy.
Okay, we'll clear the calendar, Lana.
Okay.
Yeah, Robin, Lana raises a good question, and you touched on this, the importance of of the biomarkers and of the development of a diagnostic for your group. And it gets along a little bit to what Daryl is asking about biobanks and patients and integrating clinical trials, which is something we could probably talk about for a long time. But Robin, maybe you could talk a little bit about that importance of the biomarkers and patient data in the development, and particularly for the people on this call at a really early stage. Art's very integrated in the prostate center, but how does a normal researcher really start to do that at a very early stage, do you think?
Yeah, obviously the example I gave, it's a fairly straightforward biomarker because it's actually how the disease is diagnosed. It's diagnosed through the genetic test because it's a genetic monogenic disorder. I think the industry as a whole, and I'm just going to be honest, has struggled with the utility of biomarkers that add risk, perhaps, to seeing an adverse drug reaction, or may predict that you're more likely to respond. I have to say some commercial colleagues in the industry don't like the idea of subdividing out the population through those strategies. But overall, if what's important to you is patient care, you don't want to put a patient on a drug where they're going to have a likely reaction or they're not going to respond. And I think the payers are helping us think this through, because you can now, particularly outside of the US, in Europe, for example, where you are sometimes struggling with pricing discussions, if you've got a biomarker that predicts a responder, you can have the conversation around the pricing of your drug, where you only get paid if the individual actually responds to your drug. You can command a premium pricing in that context. And therefore, having the biomarker work done during the development is incredibly important. We're fortunate about the example I gave. It's probably the simplest process for a precision medicine, because this genetic testing is done on a chip that involves several hundred genes involved in epilepsy. All of that's available to us to take advantage of. If you're developing something new and novel, you might need to start thinking about the development of a biomarker that's specific for your drug and for your patient care, the question you're trying to answer in targeting your medicine to an individual within your patient population. And you have to think about a companion diagnostic at that point. And you need to think about that early, because the last thing you want is the companion diagnostic not being available at the time when you're ready to launch your drug, because that's going to hurt you because you're not able to launch your drug in a timely way while you get the companion diagnostic together. So the earlier you think about it, the better. I can certainly see a role for AI. We actually use AI in chemistry today for our medicinal chemistry, primarily around target potency. I do think the AI technologies, and I know I'm going a little off topic here, the AI technologies need to think about more broad than just the target and binding affinity for a target. You've got to think about the other drug-like properties, build that into the system, because that's often where drug discovery programs fail. It's not finding binding affinities or finding the ability to inhibit a target. It's actually turning up with a drug at the end of the day that's suitable with reasonable de-risking to take into human beings. And that work, I think, still needs to be looked at. And that would be hugely, hugely helpful to the industry. So if Vancouver had a leading position there, that would be broadly used by the industry. But I think AI plays an important role, and there are organizations out there looking at it, is understanding the complex biology, so you can start to imagine what type of biomarkers you could look at, whether they're ELISA tests or they're more complex protein profiling tests. You need something to guide you that shows a dynamic change in the disease, which means your drug is interacting with the disease process in the appropriate way. And that just de-risks your program all the way through development, making it more likely to be successful.
Yeah, a great point. I mean, we're all trying to get to to less risky situations. Maybe I'll ask both Handel and Art, and maybe we'll go alphabetically to Art first. Talk about AI as a normal course. You've obviously done this for a long time, but how does it really get incorporated into day-to-day drug development for both academics, and then maybe Handel will talk about the experience of somebody commercializing that technology. I mean, we all believe we're going to need this tool, but what's that journey look like for everybody?
So there is a very strong, good reason why drug discovery and AI found a match so quickly and so powerfully, because you see these things coming hand in hand all the time. And that's a bit to answer to the previous sort of comment that a lot of things should be taken into account, because drug discovery is a classical multi-parameter optimization problem. And those people in the field know that. You make it more potent, then you lose stability. You make it more stable, it sticks to the albumin. You make it less sticky, it doesn't go through blood-brain barrier. And on and on and on, not to mention all these hundreds of toxicities which pop up. So you keep juggling balls in the air, and every time you have to add more and more and more, and at the end of the day, you have hundreds. So it's always multi-parameter optimization where everything depends on everything, right? And that's what the typical drug discovery and medchem nightmare is. And so AI is handling exactly this problem. So AI is the best tool to handle multi-parameter optimization. So you can pre-fine tune what you want. And we keep adding these parameters to the AI machines these days. So synthetic ability is definitely the main, number A+ parameter we have to look at, because otherwise machine can go into dreamland and come up with structures which are not doable. So we have to wire in retrosynthesis analysis. With that being said, AI is coming up with pretty tangible chemotypes, which previously people would just overlook completely as impossible. And all of a sudden, these are becoming possible and very standard building blocks too. So that's very important to know. But in principle, yes, we keep pouring in more parameters we're trying to track of toxicities, even price. So I actually published a paper a few years ago when you can predict the cost of a compound based on the structure, nothing else. So AI is amazing from that regard. And so idea is to build this generative model, which Handel can talk more about, that's his sweet spot, which will be producing a molecule with a lot of predefined dialed in properties. And it will be cooking up until it comes out. So rather than you come up with something amazing and looks nice in the crystal structure and sits with picomolar affinity, but completely unstable and gets apart as soon as it hits water. So you get something well balanced, and that's why everyone is looking into AI to make it all happen. So that's where we're going. That's what we're trying to build. Nothing is yet done, so the race is really, really on. That's where not a lot of parameters are yet put together. It's very fresh, very new field, but I can see how it's all going to come to the finish line very, very quickly. And that's how it's going to change the practice. So we will be using that for design, for prediction, for in silico tox evaluation. That's what's happening. That's what deep docking, what I developed, could be used for. You can look how this compound will be sticking to all the major toxicity troublemakers. So it's all possible. And that's what's happening exactly in the field. If that answers the question.
No, that's great. And Handel, how do you commercialize that opportunity in a world where we hope it's going to happen, and we're not quite as far to the finish line as we'd want to say in terms of proof of concept?
Yeah, I mean, that's the magic question. I mean, I guess if I could step back and just kind of baseline the discussion, when we talk about AI, it's a very large descriptor for a whole bunch of very different things that are done. It's like talking about biology, right? I mean, there are so many different branches of biology, so many problems that biology, presents and solutions, etc. So by no means is AI monolithic, excuse me, and it's a toolbox. And the problem is that when the only tool you have is a hammer, then every problem looks like a nail. That's an old adage in at least the technology business. And so, we're very careful to talk about the AI as it is in terms of what we do, and under the grand tutelage of Art and through experiences working with great organizations like Admare and others. I mean, we as machine learning practitioners have become very, very sensitive to the problems that we can solve and the problems that we'll get to them soon. And then these problems, which we have no idea. And there are going to be other companies that employ machine learning and AI to solve these specific discrete problems. Because as we all know, drug discovery, drug development, commercialization is a long process with very many discrete steps. And the further you go, the more money you spend and the higher the risk. And so we're very constrained when we look at what we do, at least as it is right now, we're focused on the chemistry. And what's been interesting is as we've moved forward and started working very, very deeply in the chemistry, we have a much better idea of it than before. We now start getting into biochemistry and pharmacology, and then, oh my God, biology and all of these things. I mean, when you take, a program into preclinical development, it's very, very different than, if you're just basically looking for hits, and we're sensitive to that. So I think the commercialization journey is multivariated. There are a lot of problems, and it's not like you can have one team that can do every single thing really, really well, at least not in a startup phase. However, what we are seeing is we're specialists in generating novel, chemical entities. We see people who are specialists in applying AI to sort of, problems in preclinical development, right? Animal models, or, modeling stability, or various other problems that we know that these are problems, but we just bolt them on to the standard process. But there are other specialists who are applying different machine learnings, and all machine learning is not the same. And so we are able to see a constellation of vendors that we can then knit together in order to accelerate the process. So commercialization being a multi-step process that we think we can do what we do really well right now, and as we continue to move forward, we start attacking these problems. But the fact of the matter is AI is, it's not a silver bullet. It's drug discovery, you need a lot of lead bullets, and we're like maybe a howitzer shell, I'd like to think, but we're still one bullet, right? And really what happens is we want to train our AI and the machine learning that we do to hit the most impact, and whether it's in reducing lead optimization, which is the longest and most expensive part of preclinical development, or hit to lead. That's an area that we're spending a lot of time on. So and these areas, as we continue to grow, will get better and better and better. And it won't just be us, but it's a whole movement, and also incumbents. Robin was talking about Xenon, employing machine learning methods. Lana, in biologics, it's a whole different world as well. These are completely different problems, and we've looked at applying our modality, or our generative AI, to various other types of approaches, and it doesn't always generalize. And so we're very good at what we do, and we focus on what it is that we do, and we want to be able to expand. But it's a complex problem. But at the end of the day, AI doesn't have to solve this magic, create a $1 billion molecule in 2 days in order to be extremely valuable, right? Yeah. It's not. If we can incrementally increase the probability of success by 10% by doing multi-parameter optimization and having the ability of a drug discoverer to move through this process and bring in common causes of drug attrition in the design of the molecule, I think kind of what Robin was talking about, then you have this unfair advantage, and it doesn't have to be like we can do it in 2 days. So I think we need to look at what the impact is going to be on a time scale as well. In 10 or 20 years, who knows? But for now, in the foreseeable future.
Yeah, I think you raise a good point. At Amplitude and other VCs, there's an investment thesis really all around that AI and compute, however you employ it, will be, as Robin said, it's just part of the drug development process. Handel and Lana's raised this as well. You guys both raised, focus on your knitting, be an expert in your own area, and yet gather that village that helps you in all these other parts of the very complex drug development pathway. And so I think I promised Jason that he could ask a question. I don't know whether he's going to come back on. Did you have a specific question you wanted to ask, Jason?
Maybe I had a comment and a question for Handel, surprisingly. I think
we're at 30 seconds, so you both have 30 seconds to get this one done.
can you apply your methodologies to a specific subset of a molecule? So varying a particular R group so that a chemist can get out of a local minimum and try to get into a global minimum?
Yes.
Yeah.
So let's talk. And Robin
Yeah.
too, I mean, I'll give you the panelist discount for Xenon. So, I'm sorry, Lana, we can't do anything on CDRs. We looked at it, but not with our method.
Well, and maybe I'll just thank all of you. I know David's going to come back on, but just in 15 seconds each, anything that you guys starting, we'll do alphabetical here with Art. Anything you want to leave the group of young drug developers with in terms of your thoughts?
Well, really, I can only mention the biggest problem of all in drug discovery, which is not science, but regulatory hurdles. So sooner or later, we'll have to face it. This is something which hopefully will be in your lifespan to deal with, but the current rate of approval is such that we are not getting even 10% drugs we need to be getting. And maybe AI will de-risk that process. So maybe AI will be part of evaluating potential hazards and whatnot, will make that process fast and cheaper. But again, you have to keep in mind that almost $3 billion figure, which typically people cite in the beginning of their presentation, that the cost of one drug, probably 2.7 billion is for clinical trial and regulatory things and paperwork. So that's where the impact of AI could be actually profound.
Yeah, no, great. Handel, just 10 seconds or so. Anything you want to leave people with?
Just to say, a lot of people on the panel here were instrumental in enabling us to get to where we are. Obviously, we're still not massively successful yet, but if it weren't for Art, if it weren't for Lana and Nancy, your help along the way, I mean, we certainly wouldn't be in a place where we are right now. So to the drug discoverers, leverage the network. And we have a tremendous ecosystem here, and people, by and large, are extremely friendly and open to helping, as long as, you're not insufferable. So that's sometimes I am. So.
Okay, Lana. Oh, you're on mute.
Building on Handel's theme, I was going to say that as academic researchers, and a lot of you in the audience probably are, it's really your job to push the boundaries of scientific exploration and publish in high impact journals. Moving from discovery into drug development and translating your drug toward the clinic and avoiding that valley of death really does. It's a whole other discipline outside of, the expertise in academia to push those scientific boundaries. So there are lots of people in our ecosystem who are here to help, who'll give you their time. They want to do it and pay it forward. We've had a lot of successful companies here that have developed drugs. So we're all here to help, even if it's on an informal basis, is what I would say. Don't be afraid to ask. We're here to help.
Great. And David has all of our emails for better or worse. And Robin, last word from the panel from you.
Sure, thank you. I would just encourage the audience, the biotech industry, it's such an honor to work in an industry where you've got such a patient-centric focus. You really want to care for these patients in medical need. And there's nothing quite like working for a company, whether it's small and you build it, or it's a larger company, and watch a product be developed, and to imagine that product being commercialized and available to patients at the end of the day. And you really are potentially improving the lives, whether it's a small number of people in an orphan disease or a large number of people in a prevalence disease. It's such a wonderful career to take on. Yeah,
it's really meaningful, and doing well will you do well is a great thing. So thanks very much, Lana, Handel, Robin, Art. Really appreciate it. And I'll turn the mic over to David.
Well, great. Thanks, everyone. And I don't think I could summarize it better than Robin just did there. It's great to be part of this community. And thanks so much to everybody, all the speakers, Art, Lana, Handel, and Robin. And a huge thanks to Nancy as well for moderating this session. I have about 20 seconds. So we value your feedback, everybody. And if you can provide feedback on what we could do different moving forward, or if there are any talks, as we move into the fall. But have a great night and a happy long weekend to everybody. So take care, and thanks again, everybody.
Thank you.